Good evening. We are going to start our lab 8 photosynthesis. Again, if you're looking at your lab book, we will be on page 93 and know your vocabulary. If we were in the lab, we would have been looking at a plastic model. Marsha will show you that on the video. So again, we do have a video for each one of these procedures, but I kind of wanted to go through them with you myself. So when we talk about how photosynthesis works in some of these, or how we're measuring photosynthesis in some of these labs that we're going to be doing, we can see the cross section of the leaf. We can see the top layer, which is the epidermis, the bottom layer, which is also called the epidermis, the palisade parenchyma, which is here. I always say it looks like a palace, like the columns in front of a palace. And then we have the mesophyll, or the spongy parenchyma, which is here. It is spongy because it's porous, and you can see all this air space in here. And so what happens is we have these stomata down here, or stoma is singular, and it is made by 2 cells, and those 2 cells are called guard cells. I had kind of mentioned them a little bit when we talked about photosynthesis, when we talked about photorespiration. So I wanted to give this a little more detail. So when there's plenty of water, that central vacuole is nice and full, and what we refer to as turgid, and so that pore is open. When the water vacuole, you see it's shrunk because it's lost water, the cells become flaccid, and then that stoma closes. And so again, what we're trying to do is we're trying to prevent too much water from going out of those pores, because we can see here, and actually I think I have, no, I don't, in this picture here, we have the oxygen that we're going to want to get out, right? That's our waste product of photosynthesis, and we're going to have our CO2 that we want to get in, but we're also going to end up getting water loss. So we want to prevent that in the hottest part of the day. That is when those C4 plants close their stomata, and those CAN plants, we will close them all during the day to prevent that water loss. So in procedure, when we look at procedure 8.1, you would be looking at the model, and then you would also be looking at a cross section of a leaf. So there would have been a slide already prepared for you. You would have gotten your microscopes out again, and you would have looked at this cross section, and you can see here we've got our epidermal cells, we have our palisade parenchyma, we have our mesophyll is all the stuff in the middle. Meso means meso, and then we have our spongy parenchyma here. This right here is a vascular bundle. This is where our xylem and phloem would be found. Xylem and phloem are where basically kind of like the blood vessels of a plant. Except instead of carrying blood, they're carrying water and sugar water. So water's going up from the roots to the leaves, and then the sugar is being dissolved in water and carried from the leaves, where it's being made in photosynthesis back all the way down to the roots, because all the cells in the plant need that food for cellular respiration. Now, the second lab that you'll be doing in procedure 8.2 it's kind of a long setup. It takes a long time to get this to work. And what you're going to be doing is taking 2 syringes, and you are going to be sucking the oxygen and CO2, all the gases, out of that spongy parenchyma. And so kind of what happens is they're not buoyant. So once you get all the gases out of that empty space, the disc that you're going to make, you're taking an old-fashioned hole puncher, single hole puncher, and punch out spinach disc. And you have one syringe that has just pure water in it, and another one that has some baking soda, basically sodium bicarbonate. The sodium bicarbonate is adding carbon dioxide to the solution, which we know plants need carbon dioxide in order to photosynthesize. So you would then be putting those syringes up in front of a light, and then as they photosynthesize, and of course both of them will photosynthesize, but the one with the baking soda is going to do much better because you actually are giving it food, basically, or at least a reactant for that chemical reaction photosynthesis to take place. And so what we can see here is as the spongy, we'll say the mesophyll, because it's the palisade, and the spongy parenchyma that are going to be photosynthesizing. If we have enough photosynthesis, we're going to get those bubbles here, right? That gas production. All right. So with that gas production, oxygen, the waste product, it will then cause those discs to become buoyant and they float. All right. So what you're going to have here is um, the disc with the carbon dioxide will float, the ones that won't. And actually some of them do. We'll eventually, sometimes we do get a disc that floats. And that's why I made sure I put a couple discs in your data here. So we do have a little bit, but you can see that, and this is some data that I've had for a while from other classes, that we definitely get more spinach discs raising up in the syringe that has the baking soda in it. All right, so you can make those observations there with that data, and then go ahead and answer all your questions. And then you move on to 8.3. Now, 8.3, we would have set up in the fume hood because the chromatography solvent is kind of stinky. And so what we would have done is you would have had spinach extract. So we would have had this green, and you'll see that with Marsha, you'll have this green pigment that you would have put on the bottom. So you can see right here the origin of that pigment. You would have put on the bottom of your paper. You would have put the chromatography solvent into a test tube, put the paper inside that test tube, and let it sit. And what happens is, as the pigments, there are 4 pigments we're going to be looking at: chlorophyll A, chlorophyll B, our xanthophylls, and our carotenoids. So our chlorophyll A and B are both greens, but one's more of a lime green and the other one's more of a blue green. Your xanthophylls are going to be more of a yellow, and your carotenoids are going to be more of an orange. All right, so I had to switch locations due to family showing up. So what you're going to do then is you're going to separate these pigments out by 2 factors. One is size. The bigger the molecule, the harder it is to move up the paper. And the second is by polarity. So we've already talked, and kind of hopefully you're starting to understand that likes dissolve likes, polar dissolves polar, nonpolar dissolves nonpolar. And so if it dissolves better in the chromatography solvent, it'll pull that pigment up with it much better than if it doesn't dissolve into it. And so we'll separate them out by polarities. So there's 2 factors that we're using to separate these different pigments out. So we can see here, this is kind of the an actual paper that I like to use. It's a really good one I always find online. But you can see here the chlorophyll B is on, I always remember B is bottom. B is on the bottom. Notice it's more of an olive green. And then we have our blue green here, which is our chlorophyll A. Our yellow here, which is our xanthophyll, and our carotene usually is like an orangish brown color. It's usually very close to the solvent line, so it carries very well with the chromatography solution, and it's small. And so I kind of have here, it kind of gives you a representation of what we're seeing with at least 3. I couldn't find one with all 4, but you can see here the difference between chlorophyll A and chlorophyll B. Now, and then the carotenoids. So the carotene, notice it doesn't have any oxygen. It is much smaller and it has no oxygen. So it is mostly a non-polar pigment, which is the solvent is nonpolar. So it goes with it it dissolves into it very nicely, versus these over here. And if you actually look at this one has 1, 2, 3, 4, 5, 6 oxygens, and this one only has 1, 2, 3, 4, 5. So chlorophyll B is more, you could think of it as more polar, because remember, those oxygens tend to make those molecules polar because they do not share nicely. All right, so you can see where chlorophyll B has a little more oxygen on it, so it makes it a little bit less dissolvable into the chromatography solution. Now, the next lab that we're going to be doing, procedure airport 4, has to do with a spectrophotometer. Now, with the spectrophotometer, this is going to be a quantitative, there's three numbers, quantity, it's going to be a quantitative lab. And the way this works is you're going to, and this is exactly what the spec looks like in our classroom, in our labs. You lift this up. There's a little slot for you to put your cuvette. This is called a cuvette. We're going to, again, put that spinach extract in it. And then we have our readings here and then our controls here. So what we're going to do is we're going to have a light source. We're going to put it through the prism. There's going to be a slit in here that's going to specify which wavelength of light we want to pass through our specimen. And then we're going to see on our little detector on the other side how much of that light was absorbed and how much was transmitted. So we're trying to see what frequency of light does the spinach extract absorb and transmit, and at how much. So this is kind of like what you would see with a plant absorption spectrum. So if we just looked at chlorophyll B and we looked at chlorophyll A, now we of course are not looking at one single pigment, we're looking at them all together, but this kind of gives you an idea that we're kind of doing we're absorbing mostly inside our blues, and then we have another little spike here in our reds kind of towards our oranges. Right, so our plants mainly photosynthesize in the reds and the blue. Now you guys are going to be doing a really funky graph. So you have your data. Your data is going to be in the video, so make sure that you either pause the video and copy down your data on page 102. On page 103 is a graph. Now do notice that graph has the wavelength on the bottom. So I particularly, when I did mine, I started and I kind of counted by fives. I started at 350 and worked my way all the way to 750. And then, kind of spread that out on your x-axis. And then, and those would be nanometers. Wavelengths of light are in nanometers. And then on the left side, you're going to have absorbance. And now I did my absorbance going up by tenths. So I started at 0, went 0.2, 0.4, 0.6, 0.8 to 1, all the way up. And I kind of separated them out again, kind of spread them out on that axis. So then what you're going to do is you're going to graph that data with the absorbance column. So you do your absorbance column, connect your dots. It is a line graph. Do it in you're going to have two lines. So do that in one color. And then a lot of my students will just color that word ABS, which stands for absorbance. There's no units for absorbance. Others make a little key so I know what color is what. And then you're going to graph a second line. And that second line, you're going to use the far right side y-axis, and that's going to be your transmittance. And on that one, I went up by 15s. I went 15, 30, 45, 60, and all the way up. And then what you're going to do is, again, you're going to use your bottom still, your wavelength, and you're going to do your dots according to your transmittance. Connect those dots in a different color. And then you can answer the questions based on that graph. So do make sure that you're able to read this graph. Not only do you need to make the graph, take a picture of it, and send it to me for homework, but you also need to understand how to read it. There will be graphs like this on your practical. So they're going to ask you how to read an absorbance and or a transmittance graph. The last piece of equipment that you would be using is called a spectroscope. Now this would be a qualitative lab. You are looking at it and making an observation with your eye. So again, we're going to be using spinach. We're going to have that held up on one side of the apparatus and send the light through it. And then you're going to look through the eyepiece. And what we're going to see when we do that is there is a prism in there as well. And so when the light goes through, what's going to happen is you're going to see when the white light goes through the slit, you're going to see the rainbow. And we have a pretty good demonstration of this in the video. So you'll see all the colors of the rainbow. When we put our specimen in front, so our spinach extract again, in front of the slit, it's going to absorb some of those wavelengths of lights. We tend to get, again, the absorptions on the ends. We tend to lose some of our blues out here, and our red band gets much smaller. And so what we're losing, the colors we are not seeing, are the colors that are being absorbed by the pigment. The colors that we do see are the ones that are being transmitted through it. So it's not using those wavelengths of light. So again, the wavelengths or the colors that disappear are being used. The colors that we see are not being used. So your homework for this lab is to complete the review on pages 105 and 106 and to do the graph. So don't forget to do your graph. Remember, again, I'm still having people turning things in late. Remember, labs are always due on Friday. I do mark off for being late unless you're lucky, and I have not graded them yet. So do try to get that taken care of and stay up to date. We only have a few weeks left, so hopefully everybody's doing okay. If you have any questions, let me know, and good luck with this lab.